Welcome to our CEDA Tech Talk series. So for the webinar today, we actually have a new co-host. This is Laura Plunkett, who is EDIH manager of CEDA. So Laura, maybe you can introduce you a bit to the audience.
Yeah, so some on the call will know my name. My name is Laura Plunkett. I'm the EDIH program manager. The EDIH stands for European Digital Innovation Hub, and it's a program that CEDAR was awarded with recently. It's a funded program by European Commission and Enterprise Ireland, and its mission is to support Irish businesses and public sector organisations to find ways to use AI to benefit their business. So really looking forward, and thank you for the invite, Kwan, to be a co-host on your series. And Dr. Dao, please allow me to see your presentation today. I'm looking forward to it, and thanks for joining us.
Yeah, okay. Thanks, Laura. Yeah. So for the presentation today, it's just about the procedure. We are going to have the presentation first, and we are going to record it. So we will leave on the Q&A at the end of presentations. And the Q&A section will not be recorded. So for the Tech Talk today, we are going to have a talk on digital transformation in agriculture with hyperspectral imaging and artificial intelligence, and by Dr. Phuong Dao from Colorado State University. So I hand it over to you, Dr. Dao.
Yeah, thank you so much, Dr. Lee. And first of all, I would like to thank you, Dr. Lee, Dr. Phan, and Mrs. Plunkett for the invitation and for the opportunity to share my research with you and all the audience in a CEDA Tech Talk at University College Dublin. My name is Phuong Dao, and I'm an assistant professor at Colorado State University. And today I would like to share with you some of my the research that I've been doing in the past few years in digital transformation in agriculture using hyperspectral imaging and artificial intelligence. So just a little bit about my background. I was born and grew up in Vietnam, and I did my undergraduate in Vietnam in surveying and mapping at the University of Mining and Geology in Vietnam. And then I moved to Taiwan, and I completed my Master of Science in Remote Sensing Science and Technology at National Central University. And then I moved to Canada to complete my PhD in Physical Geography and Environmental Study at the University of Toronto. And I have 2 years working at the University of Wisconsin-Madison as a postdoc. And now I'm here at Colorado State University as a faculty. And my research program has been focusing on understanding, characterizing and understanding the interaction between plants and disturbances, for example, drought, insect diseases, weeds and soil, nutrient runoff today, and using the integration of remote sensing, especially hyperspectral data, geospatial science, machine learning, and biochemical and bioinformatics methods. So today I would like to share with some of the research, not all of them, but some of the research I've been doing in the past few years. And as we know that our agricultural system and natural system have been seriously impacted by global warming and climate change. And the climate change driven disturbances, for example, drought, forest fire, soil erosion, pests and pathogen. And these disturbances seriously impact our biodiversity, global geochemical cycle, and also the food security. So monitoring the impact of these disturbances provided valuable information for the protection of our biodiversity and food security. And when we talk about an ecosystem, we talk about a plant first. So plant is a fundamental element of an ecosystem, and it's a complicated system with complicated functioning and metabolism, including photosynthesis, respiration, growth and reproduction, and defense and building structure for plant to grow. And in each of these processes, plant biophysical and biochemical property, also called functional trait, play a very important role. For example, in photosynthesis, chlorophyll play an important role in absorbing the sunlight for plant photosynthesis. while NSC, also called non-structural carbohydrate, play a very important role in respiration, growth and reproduction, and phenolic play a very important role in defense. So understanding, characterizing, and also monitoring the changes in these plant functional traits provided valuable information in understanding the crop health, plant health, and how they respond to the environmental changes. And these functional traits can be retrieved measures and extract using field observation or chemical laboratory extraction. However, as you know that the field observation may not be able to capture the spatial variation of the impact. For example, this is a figure that shows the impact of droughts on native and invasive species. And you can see that in this area, in the green area, plants seem to be, green areas show that when healthier, the yellow and red area. Even they are next to each other. So it means that if we use field observation or laboratory chemical assay, we may not be able to capture that variation. So remote sensing with various sensor and platform provided with very useful spatial and temporal information to understand and monitor the crop health and plant health, and especially a drone that fly very low on the ground, providing high resolution and by very high spatial and spectral resolution data. So we can have detailed information of how plant perform at the leaf level, and plant level and field level. So remote sensing, which I'd like to quickly introduce the principle of remote sensing. So remote sensing is the art of collecting, acquiring data without physical contact. So basically, our sensor that is mounted on a drone or on a satellite receives the signal that is reflected from the feature on the ground, and the data will be sent to the well observation station or receiving station for analysis. And there's many different types of remote sensing data. For example, hyperspectral, multispectral, thermal, LiDAR or radar data. But today, in my research, I only focus on hyperspectral data, optical data from 400 to 2500 nanometers. And a fundamental product of remote sensing is a digital image. It's like a digital photo that you take from your smartphone, but in your smartphone, there are only red, green, and blue RGB band. And you can clearly see that there's some distinction between different plant species or within the same species on the image. But when you look at this figure, you can clearly see that there's a lot more variation between species, or even within the same species or within the same tree canopy, because it's derived from hyperspectral data with hundreds to thousands of narrow and contiguous spectral bands. And this is an example of the spectra of one single pixel. And this, we compare healthy plant and stressed plant. You can clearly see that there's a distinction between the two spectra, a different spectral region, reflecting the variation in different plant foliar chemistry. For example, the variation in red region at 16, 17 nanometer or blue region about 450 is very is caused by variation in chlorophyll content. And the spectral from about 1000 to 2500 are sensitive to the variation in water, leaf water, sugar, phenolics, lignin, and so on. And those chemicals that you cannot see from your eye. So based on the variation in spectrum, we can understand how each of these chemical varies, each of the trait vary, and how and then we can understand how plants function. That's why a hyperspectral remote sensing is a very useful data set. And with that, we can use the data for various applications in agriculture, including crop management, resources distribution, irrigation planning, or the estimation of yield or assessment of the disturbances. And this is an example of a ternary map of the combination of leaf mass per area, chlorophyll content, and phenolics. So basically, the green, more green color showing that plant have higher chlorophyll content, which means they are more healthy. The more red color showing that the phenolic content in the leaf is high. It means that plant produce more phenolic to defend themselves when they're impacted by disturbances. So you can clearly see that in this field, you can see that irrigated field has high chlorophyll content and they're healthier than the grass area outside of the irrigation. And so this area the plant might be affected by drought, and they're not doing photosynthesis well. So they have to produce more phenolic to defend themselves from the oxidative damage or other impact. And you can see in this area as well, you can see phenolic is very high. So there might be some impact happening there, for example, drought, insect, or diseases. So you can see that this data provide valuable information for to understand how plant perform. And remote sensing can be done at very different scale and platform. For example, in here we do the lab level, field level, and landscape level. And the lab level we use a high throughput plant phenotyping system, and in the field level we use a hyperspectral sensor, and a landscape level we use hyperspectral data. And all data was processed in high-performance computing system. And so my lab is equipped with various sensors, including few spectrometer with more than 1,000 narrow spectral band, and a drone hyperspectral sensor with about 500 spectral band. We also have access to various growth chamber, experiment farm, and plant health diagnostic lab and chemical laboratory. But so today we'd like to show you some of the research that I've been doing in the past few years. But before going into detail of the application of some previous study, I would like to introduce the concept of object-based image analysis. So in remote sensing, there are two approaches. One is pixel-based and one object-based image analysis. So traditionally in image processing, people analyze or process the image based on pixel. So basically, in an image, we consider the element of the image is pixel. And in pixel, we have the grayscale as the single value that you can use for the image analysis. However, when you group the pixel with similar spectral property together to form image objects, you have a lot more information. For example, the mean gray level, the variance, the standard deviation, the length, the width of the object, and also the ratio between the length and the width. And there are a lot more other information as well. So object image analysis will provide you with a lot more information for the image analysis compared to the pixel-based method. However, one of the most important component in object-based image analysis is image segmentation, and how to determine the optimal scale. Which scale is the optimal scale when you need to segment the object and a pixel together. And in my lab, in the past few years, we focus a lot more on how to determine the optimal segmentation scale. So for example, this is we are analyzing a crop field, and this is the optimal scale. So how we determine this? So we based on we develop a method that based on the variance of the image object and the change of the variance over time across scale. So basically, an image can be segmented at different scale, and at each scale we calculate the local variance that demonstrate the homogeneity of the image object. And when we increase the boundary, when we increase the scale, then the image, the local variance will increase. And when we keep increasing the scale, when you reach the optimal scale, and if you keep increasing the scale at the boundary, the local variance will change substantially. Right? So the optimal scale is determined as the scale at which that the local variance changes substantially. So with that, when after segmenting the image, we extract the information and we input into the model for analysis. For example, for image classification or for plant chemistry estimation. And however, recently we moved one step further, that we don't only extract the feature from the optimal scale, but we keep segmenting the image further, and then to divide the object into smaller objects. And at each scale, we extract the information, we track the feature from all the scale, and we use graph neural network to integrate the information from this scale together. and then feed them into the image analysis model, for example, classification model, to improve the result and accuracy. So with that, and today I would like to introduce with you, share with you three major projects that I've been doing in the past few years, including characterization of the impact of insect, drought, and nutrient runoff on our plants. And the first project is characterizing the impact of insects on plants. So in this study, we use spongy moth as the species to conduct the project. And spongy moth is an invasive species, and it was introduced to the US in 1869. And it feeds on over 400 different species, including 300 species in North America. And it's one of the 100 most destructive invasive species worldwide. And this is interesting that in this species is that the male moth can fly, but the female cannot fly. So they can only flap their wing to attract the male. And they are spread over the space by the wind, so they cannot fly. The female cannot fly. But they're very, very destructive species. And you can see that after being introduced to the US, it quickly spread out to many different states in the eastern region of the country and across many different host plant species. So understanding the interaction between the insect and host plant species and how plant respond to the impact provided information to prevent the spread of these invasive insect species. And so to do that, we in Wisconsin, Madison, we established a common garden with more than 1,800 trees of about 520 different genotypes. These genes were collected over different latitude across Wisconsin, and the garden was severely impacted by the insect herbivore in 2021 and 2022. And I can see that we also have a competition garden, which is just about 10 kilometers away from the common garden. But this garden was not affected. That's interesting. So this insect is spread by wind. So there's no wind, or the wind direction is from here. Then the insect might not be spread out. And you can see that. This is a photo that we took on the ground. As you can see, a lot of trees or a lot of genes are completely defoliated. Some were defoliated to some extent, and some were barely infected. So we can see that there's the distinction in how these genes or these plants respond to the impact. So in this study, we want to understand the mechanism of the plant chemical defense and how one plant is more resistant to than other plants. And in Aspen, there are two groups of chemicals that play important role in chemical defense. The first is phenolic glycoside that confer toxin to the insect herbivore and reduce the herbivore digestion. So it means that the herbivore can eat the leaf, but they cannot digest, or they can die. And condensed tannin in the leaf that play important role in preventing the photooxidative damage and inhibit microbial function. And it helps the plant to alter the nutrient cycling to help them survive. So in this study, I will use hyperspectral data to quantify these chemicals and see how they change under the impact. So the objective of this study is to quantify and assess the chemical changes and explore the mechanism of resources allocation in the plant when they're impacted by the insect, and see how different genes respond to the impact. And we integrate remote sensing with biochemistry, genetic, and insect modeling. On the ground, we have an aircraft hyperspectral sensor. We have we put at the bottom of the aircraft, and a hyperspectral drone sensor. And we also collect LiDAR data using VTOL for takeoff and landing software. And on the ground, right, while you're flying on the ground, a team of about 5 to 6 people, the 10 people to collect leaf sample to measure spectra, and also to bring back to the wet lab to quantify the chemical content in the leaf. And but in the early growing season, before we do the field work, we did a biological simulation, which the model developed by Natural Resources Canada. We basically use the location, topography, and weather data to simulate and predict the emergence and the evolution of the insect. And at each, we call instars. At each instar, we evaluate how these insect herbivore impact the plants. And this is an example of the hyperspectral data that we collected. So the early growing season, May 17 was the time before the plant was impacted. And the insect herbivore emerged on June 2nd. And you can see that they gradually evolve. And in June 16, most of the plant were defoliated, completely defoliated. But there's some area and some plant that in the middle of the garden that were not impacted. And then after that, the herbivore pupate and they become moth. They were not eating anymore, and the leaf reflushed. So you can clearly see the change in the color, reflecting the change in plant foliar chemistry. And this example of a spectra that we collected from hyperspectral data from airborne sensor and from fresh leaf. So we also measure the spectra directly on the leaf, and we also flash freeze these leaves using liquid nitrogen, and we freeze dry them, grind them, and then measure the spectra of the dry materials. And then we link these spectra with the chemical data that we retrieve from the leaf in the lab. And then we use various machine learning model, for example, partial least square regression, Gaussian process regression, or support material machine regression, and to identify the spectral region that is sensitive to the variation in each of the chemical. For example, here we see that the band 20-40 nanometer sensitive to variation in condensed tannin, while the band 60 and 60 is very sensitive to the variation in phenolic glycoside. We also calculate the spectral index, and you know, in remote sensing we have normalized different vegetation index or enhanced vegetation index or so on. But in this case, I calculate the normalized indice of all the combination of all band. So which means band I minus band J divided by band I plus band J. So in total, we have maybe 100,000 of spectral band, and we link indice, and we link these indices that we calculated with the chemical data, and we can identify the area that is sensitive to the variation in each of the chemicals. And this is the result that we got. So if we measure the spectrum on dry material, ground materials, we can have an R-square of 0.98, like almost 100% accuracy. And the accuracy decreased when over the fresh leaf model and airborne model. But I mean that the result is still impressive, especially the content of these chemicals in the leaf are much less abundant than chlorophyll and leaf water. And these the map showing that these maps show the spatial and temporal variation of these chemical before, during, and after the defoliation, after the impact. And you can see that when the plant was impacted on June, about June 5th, you can see that the tannin increased substantially, and it could increase even more on June 13, right, at the peak of the defoliation. So you can see that plant in this case, when they're impacted by the insect herbivore, the leaves are defoliated. So the plant may understand that in this case we are not doing photosynthesis, so we don't produce more chlorophyll, we don't produce more nitrogen. So they don't invest more resources to produce this chemical, and instead they invest more resources to produce more condensed tannin to help them survive under the impact. In contrast, you can see that phenolic glycoside did not increase when this was impacted, and when it was very severe, the phenolic glycoside stayed the same. But after the leaf reflux, the phenolic glycoside increased substantially. This means that plant will remember the impact and increase phenolic glycoside after the impact to be prepared for the future disturbance when it's impacted by the insect herbivore in the future. And we can clearly see the distribution, right? So in this case, we can see that plant, when it's impacted, it prefer the condense and defense mechanism. That's why it increased during the defoliation. And when after the moss went away, the condense and decreased. And this figure shows the intervariation between the chemicals in the leaf, showing how plants reallocate their resources when they're impacted for different processes. For example, here you can see that before the plant was impacted, the nitrogen content is high, right? And the condensate is also high, but not too high compared to the value when it was attacked by the moth. You can see that foliar nitrogen decreased significantly, because at that point the plant doesn't need more liquid nitrogen for photosynthesis, because they are not doing photosynthesis well at that point. So they decrease liquid foliar nitrogen, and they increase the condensed tenant to help them survive. And then later on, after the impact, right, you can see that the plant moved toward the phenolglycoside, increased phenolglycoside. So it's kind of like legacy impact effect. So based on information, we can understand how plant function and under the impact of the disturbances. And in this figure, we also show how different genes respond to the impact as well. So some genes respond better than other. This is the condensed phenolglycoside and foliar nitrogen before the impact. And under the impact, you can see that phenolglycoside stay quite stable, while the condensed increased substantially. And you can see that some genes respond much better. The content was much lower than other before the defoliation, but when the plant was attacked, you can see that in this gene, the condensed increased substantially. So that is the plant that was not affected that I showed in the figure at the beginning of the talk. So this information provided would be very useful for example, crop breeding, when they want to breed more variety that is more resistant to the insect herbivore. And I'm not introducing the detail about this, but we did move one step further and not only understand how plants respond to the impact, right? How we can observe the symptom on the plant, but we also understand the behavior of insect, how they feed on the plant. This is a different species, plant species and insect species, but the method can later on be applied for insect herbivore. So in this case, we characterize the feeding behavior of an aphid on a plant. So this is an insect with a mouth pad, and it can penetrate the leaf and extract the nutrient in the leaf. And then we put a sensor on the insect, and then we have a cable that we can insert into the leaf, and then we have a sensor to record the changes in electrical voltage or waveform of the insect. And they have different phase of the feeding. For example, NP it means non-penetration. It means the mouth pad cannot penetrate the leaf. C means pathway phase, and it can mean that the plant is testing the leaf surface. And PD is potential drop. It means that the mouth pad can penetrate the leaf. So based on this behavior, we can the waveform, the change in the shape and the size of the waveform, we can understand the insect behavior that provided information on how to prevent the emergence and impact of this insect. And we use, for example, here we use random forest classification to classify different behavior, and with very high accuracy. So the next research I would like to share with you is the drought impact on native and invasive species and how these species respond to the impact. And as we know that in the real world, plant is not only impacted by one disturbances. For example, insect herbivore, but the combination of different disturbances. For example, drought-weakened plant will be more susceptible to pests and pathogens. So understanding the combined effect impact of these disturbances will be very, very important. And we know that in plant, there's plant. When you look at the plants, you can see a lot of green color, right? Because plant has a lot of chlorophyll content in the leaf, and it absorbs a lot of sunlight. And most of the sunlight will be used for chemical reaction. In the chemical reaction phase of photosynthesis, we call photochemistry, and a few percent of the active light will be converted to heat. The plant will convert active light to heat and release back to the atmosphere to protect themselves from oxidative damage. And part of them will also be re-emitted back to the atmosphere in the form of fluorescence. When they're impacted by drought, the amount of sunlight that is used for absorbed light that is used for each process will be changed. For example, it will use less energy in photochemistry, and it converts more energy to heat to release back to the atmosphere to protect themselves from oxidative damage. And so in this study, you understand how these plants interact with the drought, and how different like plant biophysical and biochemical property change under the impact. And as we know that maybe invasive species are more tolerant to the disturbances, so they perform better. And in this case, we want to see whether invasive species respond to drought better than native species. And to do that, we conducted a series of experiments from greenhouse to rain exclusion shelter and to natural drought. So we collected plant sample and grow them in the research greenhouse and apply different water treatment and see how plant responds. And in the field, we built about 10 rain exclusion shelter when we block the rainfall but allow the sunlight to penetrate. And we compare the plant inside and outside of the shelter. So the shelter had to be specially designed. So the roof is very strong. It can stay or survive under strong wind, but it can be removed and reinstalled when we need to fly airborne sensor. And those shelter were installed in different locations, different elevation and slope and aspect. And we want to see how plant perform, how plant respond over different topography. And we use in the lab, we use chlorine imaging system. And then we build a structure, and we attach a hyperspectral sensor on the top, and we have a moving platform at the bottom. So we can set up the speed of the moving platform, which means that we are simulating the airborne flight. And in the field, we use the helicopter, where we cut a hole at the bottom of the helicopter and we install the sensor, hyperspectral sensor there. And in the greenhouse, we control other factors like temperature, humidity, and light, and apply different water treatment to see how plant of different treatment respond to the impact. And as like I showed in the insect study, we calculate spectral indices, and we also calculate all the combination of the spectral band to calculate indices and identify the spectral region that is sensitive to the changes in plant chemical and to the impact of drought. And after that, we use those indices as an indicator for drought detection, especially for early drought detection. As you can see that this index, for example, radius index, that is calculated from the two band, from the 750 and 705 nanometer. And the indice can be used to successfully capture the impact of the drought after 6 days of the water treatment. But it failed to detect early stress like after 3 days. You can see that it's not, so spectral indices are not very helpful for early detection because they only use the 2 or 3 band to calculate the information. So in this case, we use machine learning model. We input the full spectrum in our machine learning model and use various different feature selection method to select the band or the spectral region that is sensitive to the drought, and use those band in a machine learning model, classification model. And you can see that from the results here, if we only use the indice, we can only achieve the early detection of about 50%, which is almost like random, 50-50. But if we use the full spectrum in the machine and deep learning model, we can achieve an overall classification accuracy of about 97-98%. can increase accuracy substantially. And in the field, we use image classification method to map and classify the image to map the distribution of these species and see how and where they are distributed in the topography. And we also characterize the topography in 6 different categories, which is ridge, upper slope, mid slope, flat, and valley. So in this topographic category represent the call it soil health or soil condition and soil water content. And you can see that smooth brome invasive species here is mainly distributed in the ridge and upper slope area because they're invasive species, so they seem to be more tolerant to drought, so they can grow in the dry area compared to native species. And then we quantify the foliar chemistry and compare them before and after the impact of the drought. For example, this is the chlorophyll content in 2016, the drought year, and this is the chlorophyll in 2017, the wet year. And this is the changes. And you can clearly see that this area, the chlorophyll content is much more stable than other areas just next to, just nearby. And we will compare with the distribution map of the species. And you can see that this area is smooth brome invasive species, golden drop, milkweed grape native species, and orchid is a native invasive species. And you can see that within the boundary here, the area that is much more stable or more tolerant to drought is actually the smooth. This is a species that was introduced from Asia, Eurasia. So maybe they're from the hot and dry environment, so they're more tolerant to drought compared to native species in North America. And we also did a large-scale study, quantify the foliar chemistry of plant under drought impact in California in 2015 and 2016. And you can see that this is the chemical sort of feature space. And when plant was healthy in 2013 and 2014, you can see that the point cloud nicely distributed at the center of the chemicals space. But when they're impacted by drought, you can see that the point shift toward non-structural carbohydrate and phenolic. At that point, the plant know that they need to produce more non-structural carbohydrate and phenolic to survive, to respond to the impact of drought to survive. And when the drought went away, you can see that the point cloud gradually recover. But it takes a few years, but you can clearly see the recovery of the chemical space. So you can see that with this information, we have this information is very useful for us to understand the impact of the drought on the plant and how the plant respond, right? So provided with valuable information for recovery and for the plant to protect our ecosystem. And the last research I wanted to share with you is my study using hyperspectral, thermal, and multispectral sensor to understand how nutrient runoff impact our crop performance. So we have an experiment site with various treatment, including nitrogen treatment, crop rotation, or combination of nitrogen or crop rotation, and drainage, water runoff. So we want to see how different soil treatment will impact our crop health and how the combination of our sensor, hyperspectral, thermal, and multispectral from the ground and from the drone or from aircraft can be used to track these changes, to characterize these changes. And first, we use a sensor to quantify the soil organic carbon, like the organic carbon in the soil. Because you know that soil organic carbon improves soil physical, chemical, and biological property, and it's an indicator of soil aeration, water retention, and water holding capacity, for example, and it play important role in soil and plant nutrient cycling and increase crop yield. So in this slide, we quantify the soil organic carbon and see how soil organic carbon changes impact our crop performance. So we went to the field and we collect data across 2 different transects over different topography, and we bring back to the wet lab and measure the spectra of this soil. And at the same time, we also did chemical extraction to extract the soil organic carbon content in the soil. And we use partial least square support vector machine and random forest to link spectra with the soil organic carbon data to build the model. And you can see that we can clearly see the differences in the spectra between the high and low soil organic carbon, right? The blue one and the red line here. And we can identify, with that technique, we can identify the spectral region and spectral band that is sensitive to the changes in soil organic carbon. And the models show that we only use the visible new information, 0.5 soil organic carbon, about 75%. It's not very high, but I think it's very impressive because this is in the soil, this is optical sensor. And then we move further to to characterize the impact of the soil nutrient runoff on our plant. And we use machine learning model to detect whether the plant is healthy or not, and when there are stress, what is actually the stress happening in the plant, whether that is a nutrient stress or it is drought stress. And we also quantify the growth of the crop over time and see how the crop grow under different treatment. And this is a crop field that we derive from our remote sensing data. So we can clearly see that the data can capture really well the performance in, especially the chlorophyll content in crop over different treatment. For example, here nitrogen and crop rotation treatment. So we will see which one is more healthy, which one is less healthy, and the level of the nutrient deficiency as well. You can clearly see that this crop field is very healthy. This one is still healthy, but it's still experiencing some level of the nutrient deficiency, while this field is completely unhealthy. So based on the information, we can provide the grower, farmer with valuable information to know how much crop, how much nitrogen or how much crop, how much fertilizer they need to add in each different field. So that's very useful in precision agriculture. And in this field, you can see that you don't see it in the field, but when you check the image, you can see the pattern here, right? It's like a striking pattern. So in this field, we dig out the soil and we insert a lot of pipes under the ground to drain out water to generate drought. And you can clearly see that the image can capture this pattern really well. And so with that, that remote sensing data, of course, there's a lot more to study, but with the advancement in a lot of sensor and platform, the data can be used for precision agriculture and various different scale, from the lab-based to ground-based and airborne and satellite. And with the help of machine learning and deep learning, we can analyze and extract information from a lot of information from the data and produce the, for example, the disturbance map here provide farmer with valuable information for protecting their crop. So the sensor, the data can be used for various different applications, as shown in the beginning. And however, one of the reason our recent study that we're focusing on is to understand how the method can be transferable across different scale, or how we can integrate the data from different scale. So basically, in current remote sensing, people just develop the model separately for the lab data, for the ground data, and for the drone data. Or they can evaluate the transferability of the methods, but they don't combine data between different scale. And if we know that the data in the lab will be more accurate than data in the field and data in the on the drone, from the drone, because you know that because of the impact of the atmospheric effect, our signal from captured on drone or satellite will be affected, and the accuracy will be much lower than field level and lab-based level. But for now, we're doing it separately. So what we're trying to in the next few years is how we can integrate and how we fuse the data across scale, how we use the accurate data from the lab and feed into the field model and drone level scale to improve our classification or our disease prediction accuracy. And also one last thing, for now we use the signal, the changes in the signal in hyperspectral data to detect the disturbances or to identify the disease. But sometimes the result that we have is not reliable. And it's because it's only based on the change in the gray level on the image. And for example, when we use spectral the image that we capture in May, then we can detect this one type of disease, for example, Fusarium disease. But this disease, I mean, biologically speaking, we know that this disease doesn't emerge in May, right? So if we can integrate with large language model with the description of those disease, we can identify that result. We can see that result is reliable or not. For example, we know that Fusarium only emerged in June and July, but not in May. So we can rule out that sort of like a prediction result. So that is one of the research direction that we are going to in future. It's to integrate image integration with the large language model to improve the plant disturbance prediction result. And once we can establish the model and when we tested it well, we can apply it to, we can transfer the method from one crop to another crop, from one plant to another plant, and from one disturbance to other disturbances, right? But I mean, this is in hyperspectral remote sensing is still less accessible compared to other data. And to do this, we need very advanced advanced system to analyze and extract information. So a lot more, I would say that a lot more to develop in the future, and it needs the collective efforts of researchers across different areas, including remote sensing, agriculture, and computer science and data science, for example. And I'm happy to be, and my group is happy to be part of the efforts. And with that, I will stop here, and I'm happy to take questions. Thank you.
Thank you, Dr. Dao.
Yeah, thanks, Dr. Dao, for a very, very interesting talk.
Thanks.
Yeah, so now I think we'll stop recording and take